Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, we'll be talking about digestive system. We'll be seeing the anatomy of the digestive system and the overview of the digestive system in this particular lecture. In the series of frequent video lectures in the future, we will see the other aspects of the digestive system as well. The gastrointestinal system or digestive system digests food and transports the nutrients into the blood. The digestion involves breaking down foods both chemically as well as mechanically into smaller components, and that can be transported through the digestive tract wall with the help of either bloodstream or with the help of lymph. Now there are many secretions are required for the digestive system to work together, along with the different muscular system, muscular actions, as well as the different GI tract and any other accessory organs. The hunger is also controlled by the secretion of some sort of hormones and other chemical components that influence the hypothalamus of our brain to control the whole process of ingestion. And not only this, hunger is depending upon the body's requirement, it's also depends on the social behavior, the social factors and availability of the food. Availability of the food in our mouth that influence the amount of food that we can eat. Now let's move on to the introduction of this anatomy. The digestive system consists of two components: the alimentary canal that starts from mouth and ends at the anus, which can be visualized here though, and the second thing are some accessory organs. Accessory organs like liver, pancreas, gallbladder and stuff. After the food is ingested, it can be processed there in stomach, which is a part of the upper GI tract, and then there will be food will be digested in the small intestine. After that, the nutrients will be absorbed and the food will be released as the form of feces through anus. The goals for this understanding is to identify the organs and circular muscle of the digestive tract, to list the structures found and represent the section of the wall of the digestive tract, to recognize the accessory organs that are involved with the digestive system, and to describe the general function of each of the organ of the digestive system, to know how they work together as a unit of organ system. Now let's look at the wall of the digestive tract. A normal section of the digestive tract reveals four main layers. If you look at here inside, one of this layer is mucosa. If you look at here, it's given here, the layer is mucosa, which is a very layer inside. Submucosa, muscularis and serosa. Serosa is the outermost layer, muscularis is right after that, submucosa is right after that, this one is submucosa, and finally mucosa, which is the innermost layer, which will be in contact with the lumen portion. Okay. The different regions of this digestive tract wall have unique structures that are related to their specific functionality. The mucosa is again subdivided into further three layers, okay, and actually three layers of different cells. Let's look at that. Those cells are known as the columnar epithelial cells because the structure is like column, so we call them columnar epithelial cells. In this columnar epithelial cells, they build up the whole area known as the villi. If you look at here, the innermost layer gets folded just like a small projections, the large folds known as the villi. If you look at one of those projections, you will see those columnar epithelial cells that make these projections. Okay. Now here, this simple columnar epithelial cells, they are densely attached with each other along with some other cells known as goblet cells, which are some sort of secretory cells which start secreting mucus. A lamina propria is another region. If you look at here in this picture, it's written clearly. Lamina propria is a connective tissue layer that contains the blood as well as the lymphatic vessel, which kind of reaches to the top of this projection. So this is the lamina propria connective layer, contains the blood vessel as well as the lymphatics. And there is also a smooth muscle sheet called muscularis mucosa that is present in the base of all this structure. The mucosal epithelial normally functions in both secretion as well as the process of digestion of the substances, and it also involved in the process of absorption of nutrients. Here is the view of individual cell is given. You see, these cells are filled with so many sac-like structures, especially they have Golgi apparatus inside, vacuoles inside, because they are designed to absorb nutrients. Goblet cells that are present here, these are the goblet cells. These are secretory cells that start releasing mucus, okay, which is nothing but a hydrated mucin protein. They produce mucin proteins, they hydrate that, it converts into mucus. While the other mucosal epithelial cells secrete digestive fluids, as well as they have the property of absorbing the nutrients. There are other type of cells nearby to the goblet cells also. It's known as enteroendocrine cells. And these enteroendocrine cells, they start producing hormones. That's why they're known as the enteroendocrine, because they're enteric cells. Let me take a color. I should explain it with color. These are the enteroendocrine cells. These start making hormones that are required for the process of digestion. It's not exactly for the process of digestion process, but it also helps in the hunger control and a lot of other control issues of the digestive measures. Nutrients which are absorbed by the epithelial cells, they either move to this capillary or this lymphatic and then travel the rest of the part of the body. These mucosal epithelial cells are also mitotically active, so they can divide with the help of mitosis, cell division. So this structure is not permanent, it's not fixed, it may change. It actually gets changed almost like every 3 to 6 days. They produce new cells to replace the old cells, because those cells have continuous work of absorbing and the process of digestion. Okay. Now if you look at here, this muscularis mucosa, that is the layer. It's a very interesting structure actually. This muscularis mucosa have a double layered structure, and this double layer is to aid for the digestion as well as for the absorption by moving the mucosal villi. So this muscularis mucosa, you can't see it here. If we go back here, you will see the muscularis mucosa. This is the muscularis externa. Muscularis mucosa is this layer. This is the layer muscularis mucosa just in the basement of the villi, and it is attached to the villi. So if I draw muscularis mucosa here, it's attached to almost like attached with the villi. This muscularis mucosa have two different layers, and with the help of these two different tissue layers, it can actually move the villi in different directions, different orientations. This movement or wagging like movement, this movement can actually help this villi to interact with many different nutrient molecules at the same time, and so that it can absorb nutrients that can travel through this blood vessel and lymphatics. And there is another layer, remember here, muscularis externa. It contains again two sheets of muscle. One is a circular, another one is a longitudinal layer. Two types of layer you can see. That's why you see these ring like structures in the middle. Now this is the structure. The fibers in the two layers, they arrange in the right angles to each other, and they provide structural support and as well as elasticity to the intestine. And this elasticity is very important for a process known as peristalsis. Okay, peristalsis and segmentation are produced by the contraction of the muscle of the intestine, And actually the muscle, if it's only one muscle, longitudinal, this kind of contraction is not possible, because peristalsis means it will be a close contraction of the muscle. As you see, close contraction of the muscle in nearby areas that allows the food that is present to move forward to a specific direction. In this case, the food will be migrated from the esophagus, from the mouth towards the anus. That is the direction of the food movement. So we need both type of tissues. We need the longitudinal tissue as well as the circular tissue for this whole process to work. Okay. Now another specific region, if you look at it, this picture, you can see it is myenteric plexus. Myenteric plexus is a network of neurons that is present in the muscularis externa. If you see, it is very close with the communication with the submucosal plexus as well, and together with the two plexus they produce the enteric nervous system. And this enteric nervous system is very important, because whenever the food is moving, our intestine should crosstalk between the brain and the intestine, and this crosstalk is mediated with the help of this nervous system that is actually present in myenteric plexus. The outermost layer of the digestive tract is the serosus, as we talked here, the serosa, right? This is also known as visceral peritoneum. Peritoneum is a outermost layer always. So this is a serous fluid producing serosa, which actually lubricates and lubricates this whole intestine, and it reduces the friction of the digestive tract within the ventral body cavity. So it starts releasing all those lubricating compound outside. Now this whole component of digestive tract, it is present in our body cavity, right? So there could be like friction between the other area of the body cavity, ventral body cavity actually, with the help of this with this digestive tract, because once you eat things, once you drink a lot of water, the area is going to increase in volumetric size. So it may interact with the ventral portion of the body cavity, and that could be dangerous, that could be bad for our health. That can be prevented by the serosa. Now let's talk about the upper part of the gastrointestinal tract. The upper part of the GI tract involved with the process of ingestion, because it starts with mouth, ends, the upper part almost ends at the end of the stomach. That's what we call the upper part of the or upper GI tract. Food start ingestion in the mouth, then inside the mouth chemical digestion take place, because there we have saliva, and saliva contains alpha amylase enzyme, which can break down starch and sugar. Okay, and also mechanical digestion takes place there, because we have teeth. With the help of that, with the help of teeth and tongue, we can break those food down into soft round saliva mixed content known as the bolus. Okay, you see the lining of the oral cavity and the pharynx is mostly made up with a specific type of squamous epithelial cell known as the stratified squamous epithelial cell. Okay, stratified squamous epithelial cells, actually squamous epithelial mucosa, because all this esophagus line is covered with mucus to prevent the damage by the acid reflux. The partially digested bolus, because we can say the partially digestion of the bolus, because it's mixed with amylase. This partially digested bolus of food is then moved from the mouth to the esophagus. Esophagus is the connective tube between the mouth and the stomach. Okay. Now the transition of esophagus wall is slightly shown here, from the start striated to the smooth muscle. The striated muscle are present in mouth region, but now we have a more of a smooth muscle, but its component of this esophagus is both striated muscle as well as smooth muscle. But when we go down to the stomach, you will see most of them made up with smooth muscle, completely, mostly made up with smooth muscle. And when the food enters stomach, it's involved with the chemical digestion, and the digestion take place in the stomach are mostly for proteins, okay, and a little amount of mechanical digestion takes place as well. So these are the three major components of the upper GI: mouth, esophagus, this one, and stomach. These are the three components. Now let's talk a little bit about the stomach, which is one of the most important constituent of the alimentary canal. The stomach, if you look at the structure, the stomach is divided into 4 different sections. Starts with cardia, the beginning of the stomach, fundus, just after the cardia, body, which is the most of the part having a huge surface area, and pylorus, which is the end point of the stomach where it interacts with the duodenum. Now if we look at here, there are different layers of muscles present. We have 3 types of layer. We have a longitudinal layer, you can see it here, straight lines, circular layer wrapping around the stomach, an oblique layer which is a linear structure but not like the longitudinal. 3 different layer of muscle is present, and these layers are very important for maintaining a tension and for fitting to the stress that is going to occur in the stomach, because stomach will contain the food, food will stay there for a while, and it will do the mechanical shearing of the food. Now here you can see, if you look at the muscles from the outermost side, what we can see is you can see that muscularis externa layer, that we can also saw earlier, that muscularis external layer of the stomach, it is unique because it carries these 3 different layers. And another very important structure of the stomach is this kind of folds, the inner layer folds of the stomach. Due to these folds, the stomach can increase its surface area, increase its size based on the amount of food it receives. Okay, these regions are known as rug, G-U-E, yes, sorry, G-A-E, rug. Rug are specific cleaves or this kind of folds that are internal folds that we saw in stomach. Okay, so stomach can expand due to its requirements. Once the food is mixed with the gastric juices which are secreted by the stomach itself, the stomach is called all this food mixed with the gastric juices is known as the chyme. Remember, it's known as the chyme, which is then moved from the pylorus, which is this point, the end of the stomach, to the duodenum, into the small intestine, because this is the end of the upper GI. Once the duodenum starts, we call it the lower part of the GI tract, and that is the ultimate job of the food to be removed from the pylorus to the duodenum. Though there is a specific valve present known as the pyloric sphincter, we will talk about that later. Let us move to the second part of this intestinal tract, that is the lower part of the GI or lower GI. Now the second part of the gastrointestinal tract is the lower GI tract or the lower part of the gastrointestinal tract or alimentary canal. It mainly composed of 2 different units, one of which we are seeing right now. First unit is the small intestine, and second is the large intestine. Okay, pardon for bad handwriting though. Small intestine and large intestine. Small intestine is a portion mainly, and actually if we combine both of them together as a lower GI tract, the majority of the chemical digestion process and actually all the nutrient absorption process take place in the small intestine. Okay, large intestine is the portion which carries the last undigested materials, convert them into feces and help them to be defecated outside. Okay, now if we look at the small intestine, this slide is about that small intestine structure. Small intestine is divided into three different parts which are illuminated in this picture with this yellow, purple and pink color. Starts with duodenum, yellow color, jejunum, color is purple, and ileum, colored pink here. These are the three different regions of the small intestine. And if we look at the structure of the small intestine, it's very much similar that of a structure we saw in stomach as per the tissue layer or histological structure proceeds, but a little difference as well. You know, in this case we also see three different layers in the inner wall of the small intestine and three different layers of the tissues, longitudinal, circular as well as the third layer. But the unique feature about the lower intestine, the small intestine, is to have a finger-like projection known as villi or villi. As you can see here, you see this finger-like projections all around this internal surface of the small intestine. This villi is to increase the surface area because the job of small intestine will be to absorb nutrients. So the more surface area they have, the better absorption they can do. Okay. Now this villi, if we zoom into villi, one of those villi, you see the villi is further divided into small fragments. This smaller, even smaller fragments are known as microvilli because these are the constituents of the villi, right? That is the part of the small intestine. And this intestine aids the body for defenses against different pathogens by secreting antimicrobial compounds as well as antibodies, which are also known as immunoglobulins. Those immunoglobulins which are produced there in small as well as in large intestine, they also produce those antibiotic compounds that can fight against pathogen. Though in the large intestine a lot of healthy good bacteria resides, and that is very, very important for our living. Now let's look at the large intestine, which is almost the last part of the alimentary canal. It also consists of several fragments. It starts with ascending colon, transverse colon and descending colon. These are the three different parts that we see here. Okay. The job for this large intestine is to hold those undigested foods, do a water absorption, and then finally conversion of those undigested food into feces, and then release them through anus. And the partial undigested foods, they will store, the feces will store for some time in the rectum. And there is another unit called cecum, which is attached to a specific portions, which is an extended region known as appendix, the function of which is moved now, that means which is not required now, but somehow in the evolutionary history they have some function in the digestion, they played some digestive role. Now here you see in those large intestine as well as small intestine, what they have mainly in regions of ileum, in the small intestine they have lymphoid nodes. In the ileum they have lymphoid nodes, and those nodes are known as Peyer's patches. Peyer's patch. Those Peyer's patch are filled with leukocytes. Leukocytes means white blood cells. which are the immunogenic cells. They can help to fight against infection. Okay. Now, in the large intestine's job, besides the storing of the undigested food and convert them into feces, the other job is to absorption of water and salt, and also absorption of vitamin K. There are also other bacteria present there. The healthy bacteria I talked about, they start producing a lot of vitamin K and then supply the vitamin K to our body. The large, as you see, there are 3 bands of smooth muscle again called the tinea coli. It's caused the outer portion of the colon to be packed into the pockets called haustra. That is the structure. That's a unique structure though. Haustra is a region where they are properly embedded in the body so that it's remain attached with the outer portion of the body. Otherwise, rest of the portion of this digestive tract is not linked in any way. Okay, they're separate from the rest of the ventral body cavity. Okay. Now we will see some other accessory part of the digestive tract that also helps in the process of digestion and absorption of the food. One of such thing is sphincters. Sphincters regulate the passage of food from one region of the digestive tract to the next, and then finally it helps for the feces to get out of the body. So the role of sphincter in the process of controlling the digestion is huge. As you can see in this left hand picture, the sphincters are present every different branch points of the digestive tract. Here you see the sphincters of the digestive tract divided into mainly 6 different sphincters we can see here. One of the sphincters are related to esophagus, known as the esophageal sphincter, known as the upper esophageal sphincter, UES. Okay, and then lower esophageal sphincter, LES. Okay, both of them are physiological sphincters. Then at the end of the stomach, if you see here, at the end of the stomach, the junction between duodenum and stomach and the pylorus of the stomach, there is a sphincter that controls the release of the treated, mechanically and chemically treated compound in the stomach to take entry into the duodenum. Okay, there comes the role of the pyloric sphincter. And then it comes down to ileocecal sphincter, which is a circular smooth muscle type of cell. It's not any kind of physiological structure. It's a smooth muscle cell in circular fashion. Both this pyloric sphincter as well as ileocecal sphincter. Ileocecal sphincter is also found, and you see here ileocecal sphincter found in the interaction, the junction between the small intestine with the large intestine. So as I mentioned, these sphincters are present in all of the branch points. And then we have internal anal sphincter and we have external anal sphincter. Both are present in the anus area that controls the movement and release of the feces outside the body. Okay. The upper esophagus sphincter prevents the air from entering into the esophagus. The lower esophageal sphincter prevents the acid reflux, which can go from stomach to the esophagus, that can cause the damage to the esophagus, or acidity is the prime problem with this acid reflux. Pyloric sphincter, this one, regulates the passage of chyme from the stomach to take entry to the duodenum. The ileocecal valve controls and regulates the passage of chyme from ileum to the large intestine. And then the last 2 types, like the internal and external anal sphincter. The internal anal sphincter, or IAS, when it is relaxed, it produces the urge to defecate. When it relaxes, defecation occurs. And external anal sphincter, when it relaxed, it allows for the defecation. So both of them needs to be relaxed for the process of defecation. Now let's move to the last part of the discussion, which are accessory glands. There are several accessory glands which are not directly involved with the gastrointestinal tract but plays very important role. One of such is salivary gland. We have normally three types of gland that are important: salivary gland, liver and pancreas. In the salivary glands, they moisten food, cleanse and protect the mouth, and it produces alpha amylase known as a salivary amylase, because the same enzyme is also secreted by pancreas known as pancreatic amylase. This amylase enzyme helps in the digestion of starch and other carbohydrate molecules. It can break down starch. The second one is the liver. This is probably one of the most important organ of all. From the perspective of digestion itself, it is very important because it produces bile, which actually emulsifies the fat, because without the help of the release of this bile or bile salts, we cannot be able to digest fat, because this bile neutralizes that by emulsifying the fat molecules and break them down and increase their surface area, so that other lipase enzyme can actually break down those lipid molecules. Now this bile that is secreted, sometimes the extra bile that is released by the liver is stored in a small sac nearby the liver, which is attached to the region of the duodenum area. That part, that small area, small section is known as the gallbladder, which stores this bile. Okay. And then third one is the pancreas. The arrangement of liver, pancreas, gallbladder is very close to each other, and they are all connected because they have a simple single duct system through which all those necessary enzymes are released into the duodenum. Here you see in pancreas, they have a main proteolytic enzymes to be produced. It releases different host of digestive enzymes into the duodenum with the help of the same pancentric, the same type of duct that we use. The duct, when it's involved and engaged inside the pancreas, is known as the pancreatic duct, but that is the duct has the branch and attached with the gallbladder as well. Okay. It also produces bicarbonate. Bicarbonate can neutralize the acidity of the chyme that is released right after the treatment in the stomach. That is another important priority, to make the chyme neutralized before using them in the intestine. Otherwise, intestinal cells may have some problem with this acidity. Stomach cells are designed to sustain that acidity. So that, in a sense, are the anatomical overview of the digestive system. Welcome back, friends. This is the second part of our discussion regarding the digestive system and the digestion of food by human being, and this is about the motility of food during the process of digestion. Motility is the movement of food through the gastrointestinal tract, and the muscles of the digestive system and digestive system tract walls are also very responsible for propelling and mixing those foods or food contents together. Let's see the whole process in details. The goals for our learning is to discuss about the types of motility found in each section of the digestive tract, to describe the function of each type of motility, and to describe the control of those motility in the digestive tract with the help of two things, either the nervous system or the hormones. The start point of this whole process of eating a food begins with chewing. Chewing is a mechanical digestive process that tears down the food, grinds the food into smaller pieces and enough small for swallowing them, because the area through which the food will enter into our GI tract from outside into the esophagus are very small. So for that to happen, we need to break the food down. So there are two processes going on there in the mouth. One is the mechanical breakdown and another is the chemical breakdown. That's very little part, though the main part is the mechanical breakdown, and chewing is one of the situation. Chewing is one of the procedure of the mechanical breakdown. Chewing also mixes the food with saliva, and when it mixes the food with saliva, saliva contains amylase. That is called salivary amylase, that can break down starch and the polysaccharides, which is the sugar part. Chewing also have both type of action and control and reflexes, like voluntary as well as involuntary reflex components. The small intestine reflexively slows down this gastric emptying to allow the neutralization and the enzymatic digestion and absorption of its contents. Okay, so that we can take some more food and break them down and then engulf it inside. Okay, the second part, after chewing, the food start to move through the esophagus and ultimately it will reach the stomach, and this whole part of entering the food from the mouth into the esophagus there is known as the swallowing. Okay, and in this case, esophagus has the responsibility to move the food that is known as the bolus, which is the chewed or grinded food, and they take that food from pharynx to the stomach. So that means pharynx means the place of the throat to the stomach, where the food will reside for a while and then it will be broken down. Protein digestion will take place a little bit there. The esophagus also moves its content via a process known as peristalsis, that we talked about earlier in the first part as well. Peristalsis is like a kind of a closed contraction of the muscles that will allow the food to move. Like it's like if the food is present here, you'll squeeze the muscle here, so it will push the food towards a specific direction, and this direction is towards the stomach. Now swallowing begins voluntarily, but it is completed by the reflexive activity, and reflexive activity is involuntary in nature. Now there are two types of peristalsis, primary and secondary. The primary peristalsis controlled by swallowing center in the brain. There is a specific center for that. And the sequence of events that happens in swallowing step by step is, the first part is a soft palate rises to close off the nasopharynx, because there are different kinds of valves present that prevents the passage to the nasopharynx, the nasal passage, as well as other part is like, because the nasopharynx and the nasal passage as well as this GI, this esophagus, they are placed very close to each other, and if a part of the food particle enters into the nasal passage, it can block our airway, that can lead us to the death and choking. So to prevent that, there are valves that will close up that nasal passage, and that's what happens at the very beginning. And the second step is the tongue start to rotate, and it try to start to force this bolus, which is a saliva mixed grinded food. It will force the bolus to go into the first area of the pharynx known as the oropharynx or oropharynx. Now the larynx elevates and it will start closing the glottis. Glottis is another part of our nasal passage through which it's added there. So the glottis will be closed there afterwards. And after the closure of the glottis, the epiglottis covers the glottis. So by that fashion, the complete nasal passage is blocked. So if you think of that, this food is moving from this mouth and there are two different passages side by side. This is a food passage, this is a nasal passage, and this is the esophageal area. And there are glottis that will be covered, and then epiglottis will cover that up. So ultimately the nasal passage is completely blocked. So now the food can easily move through the esophagus only, right? So now the pharyngeal muscle start to contract, and there is a specific valve known as the upper esophageal sphincter, or UES, upper esophageal sphincter. There is a sphincter muscle that present in the joint between the esophagus and this passage of our mouth, and that is the area this upper esophageal sphincter start to relax, that allows this bolus to take entry to the esophagus, and then the primary peristalsis process will start to begin. Once the food particle enters, once the bolus enters inside the esophagus, then the process of peristalsis begin. And the peristalsis is a consecutive wave, like this is, if you imagine, like this is the normal width of esophagus, then it will be contraction of the muscle side by side regions, and that contraction will allow the food particle that is present. These are the contraction regions that will allow the food particle. Let me change the color here. I don't know, I'm not getting, 1 minute. Let me take that another color, let's say red, let's take black. So in this case, this close contraction will push this food to the direction of the stomach. Okay, that's the process, and then the bolus moves to the stomach, and finally to the end of this whole esophageal valve there is another sphincter, just the junction between the esophagus and stomach, known as lower esophageal sphincter, or LES. And once the food take entry through the LES into the stomach, the LES will close down. So the stomach, it will look something like this, like if I draw it like that, there's a sphincter. So once the food particle take entry, they will close up that area of the tissue. So now the stomach is completely separated, the esophagus is blank, and there is a valve in like closed, so that the stomach contain the food only there. Now the larger the size of the bolus, they start to make more force, and the more force should be made, and it should be generated by the peristalsis in the esophagus to push it to go into the stomach. And it kind of take approximately 9 seconds for a typical bolus to move from the esophagus to the stomach. So it's very, very fast. It's only 9 seconds for this whole process to complete. Okay, and like liquids, water, and for these things it will take only about 1 second to travel. That's very fast, because all the process we discuss slowly, step by step, but all these things occurs very, very fast, simultaneously, to do this whole process. Not simultaneously, actually sequentially mostly. Now once the food is in the bowl, like in stomach, as you see here, this is the where the picture says us about the peristalsis. Like there is the bolus, and the muscle contraction just above it, it will start creating a force and pressure to the bolus, and the direction of the pressure is towards the stomach. So the food particle will start moving, and ultimately it will reach the stomach through the lower esophageal sphincter, and once the food particle reaches stomach, that sphincter closes down there. Okay. Now the relaxation as well as the contraction of both of these are very required for the peristalsis process to continue with it, that we will see. The relaxation of the peristalsis that we require, that the stomach normally stores food, and it mixes this food with the gastric juices for some sort of chemical digestion. Mostly the digestion take place for the proteins, and then they will empty all those contents into the small intestine, that is the first part of intestine known as duodenum. Now in this case, repeatedly like relaxation of the specific area of the stomach known as the fundus are required, as well as the structure called as body, because this is the part known as the fundus, this is the part known as the body. So fundus, body, these are the parts should be relaxed in some extent to allow approximately 1 liter of the food to enter into the stomach. So once you are looking at a stomach in the emptied condition, you can't actually believe how much bigger it can be. Once the food particles start to enter inside, the fundus and body start to relax, it can allow up to 1 liter of the food to be stayed inside. Okay. Now the peristaltic contraction of the stomach also take place, and that is also required for pushing the food through the stomach into the duodenum. That's also very required. Like it occurs like every 3 to 5 times in a minute. it mixes the stomach content with gastric juice and empty the whole content, which will be known as a chyme. In this case it will be known as a chyme, because once the bolus take entry to the stomach, it's mixed with all the acids and all the chemical components from the stomach. It's known as chyme, and then the chyme will be taken into the duodenum from the stomach with the help of peristalsis. And the frequency of this peristalsis contraction, it is regulated by the interaction between the pacemaker cells that are present in the smooth muscle cells in the stomach wall. And larger volume of food the stomach is getting, it's to produce a stronger gastric contractions, and the smaller volume of food leads to the less gastric contraction in that sense. As you see here, the peristaltic contraction begins from the first point, and once it start to push this food particles to the downside of the stomach, then again further peristaltic contractions start to occur and it will push the food to the duodenum. Okay, and there is another sphincter that is present between the stomach and duodenum junction, known as pyrolic sphincter, that also regulates whether once the food enters into the duodenum, it should not come back into the stomach again, because it's not required in that sense. So here is another picture explaining the whole process of the peristalsis that goes on in the stomach. It's approximately 3 to 5 times per minute. But still this peristaltic contraction is really important to push the food. If it's a more solid food, then the contraction is even further, even stronger. Now this whole process of contraction is regulated by our brain, different centers in our brain that actually gives the idea whether the food is large enough, so we need to generate a larger force to put it into the duodenum and stuff. Now if you go here, the chemical composition of the food as well influence the rate of the digestion. For example, if it's a lipid rich content of our food, in that case it will take much longer time to digest than the carbohydrate rich food, because carbohydrate rich food have a digestion started from the mouth and the whole process, and a little this whole, because the carbohydrate can be digested with the amylase that's secreted there, and the amylase will be secreted from pancreas as well. Lipids are much hard to work with in the digestion process because they are not water soluble, so we need to break them down in smaller fragments with the help of bile salts. Then the rest of the process we need to do sequentially to finally take it inside our body. But in this case, the contractions of the antrum that propels the chyme into the duodenum is small squirts that happens here at the end of the junction of the stomach and duodenum, and there is a sphincter in the middle. And in this case, the whole process starts to work like small squirting size, one by another. Okay, for example, and this is whole process of squirting foods like small amount of food at a time. It's known as the gastric emptying. Gastric emptying. Okay, that's the release of the food product or chyme from the stomach to the duodenum, and they transfer it through the pyloric sphincter that works there. Now the closure of this pyloric sphincter forces the chyme to back into the stomach for further mixing. So what happens here is just like mixing everything, putting a little bit to the duodenum, again it closes down so that it forced back to the stomach again, then again it try to push it to the duodenum. So this is not a one-time event process. This process is working slowly, once at a time. A little amount of food is transferred at a time from the out to the next. So for digesting a yogurt, it will be much faster than digesting all these components of this cheese content and a lot of chicken and related content. So that is another thing that you need to, this is a slow digestion, this is a fast digestion that we will see. And the rate of this gastric emptying is also slowed by the presence of fats, acids, and different kind of hypertonic solutions that is present in the duodenum. Okay, the different types of regulation of this gastric motility that we want to talk about. Remember, we already talked about it, that there are two types of control. One is the nervous control, another one is the hormone mediated control, that's the signal mediated control. Now in hormone mediated control, that is the most of the part of control that we will see here in this case. But normally nerve also plays a very important role, because nerve controls the response of the stomach during the cephalic phase of the digestion. That's called the cephalic phase because that phase is controlled by the cephalon, that brain content. The cephalic phase is regulated by large long reflexes, and especially the vagus nerve. The vagus nerve regulate the phase of this long reflexes in the cephalic phase. Okay, and this vagus nerve carries the signal from the brain to the stomach. Remember that it carries a signal from brain to the stomach. It's a large nerve, and during the cephalic phase, and both the nervous and the hormonal control the whole response of the stomach and how the stomach will empty all those gastric contents and chyme into the duodenum and the whole process. Now if you look at the process of hormones, you can see the gastrin is the hormone that regulates the gastric secretion, and this is the role of gastrin. It acts as plus, means it's going to the positive effect. So gastrin is secreted by the cells present in the stomach, and that acts on the stomach to release more of those gastric juices. And stretching the intestinal wall stimulates the stretch receptors, and that cause the response by the duodenum, while the gastric motility and emptying decrease as the duodenum begins to receive chyme from the stomach. That's another important thing to happen. Okay, so as it start to receive the motility from that point, it start to slow down a little bit, and the fats in the duodenum causes the release of another hormone, CCK. If the fats are present here, it stimulates. This is known as cholecystokinin. That also stimulates gallbladder. CCK stimulates gallbladder to release bile salts, because the biles will be required to break down fat in smaller fragments, so that it can actually separate fats in smaller fragments, which is called as an emulsification. This emulsification is required so that, like, if it's a large fragment of fat, it will break the fat down into smaller chunks so that it increases surface area, and so that other lipase enzymes can work to break it down in smaller fragments. Okay, now both the hormones secretin as well as the nerves elicit the response that helps the duodenum to contain those acidic content inside. And different hypertonic solutions in the duodenum, as I told you earlier, it also elicit a response, but the precise mechanism is not fully understood there. So secretin is released from here as well, from the duodenum, and that will act on pancreas to start releasing different pancreatic enzymes like pancreatic lipase, pancreatic amylase, pancreatic different proteases that will help in this whole process. So there is a cross interactions one by another for this whole process to interact with themselves. But here the automatic communication between the intestine and the stomach is called enterogastric reflex, and that's a very interesting thing, that how the stomach and the rest of the intestinal region interact with themselves with the help of all this hormonal signaling process, and very much well coordinated. Even we can't feel any of this when we're eating things, because once we're eating, we're enjoying the food. After the eating of food, we are sitting, we're watching a movie, or we are working, so we don't even hardly think of what's going on there, because it's not bothering us much. But this process is going on every single time we take a food inside our body. Now if we talk about the migration of this food during fasting, what will be the case? Now we talked about when the food is migrating at the very normal constituents, when we are eating food consistently in a small amount throughout the time. That's the normal process, that's the healthier process of eating a food. But if we like after a meal is digested, during the interdigestive period. Interdigestive period means we have a consecutive to digestion and there is a gap between the two digestion. The segmentation is replaced by the migration or motility of the complexes that are present involved with the digestion of the food. Migrating motility complexes are nothing but peristaltic webs from stomach to the ileum what sweep the undigested material towards the terminal ileum portion. That is kind of fasting. So if you do a fasting, it kind of results in putting all those food that is present any part of our digestive tract to ultimately get to the clearance part. That's the end point of the ileum, which is at this point. So it's something like brooming. So that is why fasting sometimes is required in little amount as well. And the frequency of this migrating motility complex, it's known as a migrating motility complex. The frequency of this migrating motility complex is kind of a once every 90 minutes or like 6 to 8 overnights. So 1 in 90 minutes you can say, like it start to push all those things, whatever thing is present, whether it's digested or not digested, it will drag it to the end part of the ileum. Then it will take entry into the large intestine and it will convert into the feces and it will come out. Now this whole process of migrating motility complex are controlled by enteric nervous system, and we don't want to talk about that much here, but that's how it's also important to clear all this route for the later stage of digestion and stuff. Let's talk about the stuff about segmentation in a little bit more detail, because we heard this name earlier as well. There are two major functions of the large intestine that we know of. One is the storing and concentrating the fecal matter. So fecal matter storage, and the second thing is absorption of water, salts and vitamin K. That is another very important component. So here you know that between the ileum at the very end and the beginning of the large intestine, there is another sphincter known as ileocecal sphincter. Let's say here it's the small intestine. So ileocecal sphincter is present here, and this ileocecal sphincter opens to allow the chyme to enter into the cecum. Then this sphincter gets closed, so that once things are entered into the cecum, the sphincter is closed, and that will allow to block the backflow of this material. Now then this chyme, approximately like on average it kind of like 500 of this junk materials per day this whole large intestine needs to take care of and release outside the body. Now here where we require a process called segmentation. Segmentation is a structural separations. It's not fully separation but partial separation to form a partial compartments in the large intestine. And this tiny segmentation that we see here, the partial segmentation that we see, the pouches that formed here known as haustra. It's known as haustra. As you see, this segmentations here. These pouches are formed and it's actually formed in the transverse to descending colon. Till descending colon we find it, like from here cecum, then transverse colon, then till descending colon. And the mass movements are sustained and intense peristaltic contractions start to begin in the large intestine due to the formation of this haustra that we see. And the peristalsis will start from this point and it will slowly start pushing the food through the transverse colon till the descending colon there. And this haustral contraction and mass movements propels the feces towards the sigmoid colon and finally into the rectum. And this movement of peristalsis is not from a one single direction, as they have multiple segments. Segmentation plays further increased role in the process of clearing all those fecal materials. As you see here, if there is only one kind of simple primary peristaltic wave start from our end, it will push all these materials towards the direction slowly. But as we have a higher segmentation and contraction of all those muscles, that will allow the food and all those fecal particles to move down this whole process and till the rectum very fast. Okay, and the destination of the rectum causes the internal anal sphincter. There are two types of sphincter present: internal anal sphincter and external anal sphincter present consecutive area. Both of them needs to be relaxed for release of the material. So in this case, the internal anal sphincter have to relax, which produces the urge to defecate, and this is involuntary smooth muscle, while the external anal sphincter is kind of a skeletal muscle which is under voluntary control. Otherwise it would be very difficult for us to live in this world, right? So in this case, both the relaxation of internal as well as external anal sphincter is required for the urge for the defecation. Now, urge of the defecation is developed by the relaxation of internal anal sphincter, while relaxation of external anal sphincter will actually make it happen, and that is under our control. Only about 150 ml of the 500 ml chyme that enters into the cecum is eliminated as feces. So it's kind of says this is the percentage. It's not even 1/3. Approximately it's kind of 1/3 percentage that goes, and most of the remaining 350 ml it is absorbed as water, because that is the portion, you know, the whole part that take entry, the 500 ml components, that 500 ml components are carrying all the materials, digested materials as well as the unwanted undigested materials. So the body will take and absorb the digested nutrients, and it will start to defecate with the undigested materials over there. Okay, so feces contain mostly undigested foodstuffs, and it will also consist of bacteria, especially E. coli. There's a dry weight in the feces. You'll see 1/3 part of the dry weight is only with E. coli bacteria. So you can see it's residing there for a while, and they actually provide vitamin K. They produce vitamin K, they provide the vitamin K to the host. So this is kind of a beneficial to have E. coli in our gut. Now about the absorption, I told you here, the absorption that the intestinal cells, the large intestinal cells are involved with is the uptake of sodium, chloride, these ions, as well as it will absorb water, a lot of water. And here are the cells we use different type of transporters, co-transporters that will allow the sugar, and like one example if I give up about sugars, to take sugars inside with the help of a sodium density gradient. So it's kind of a co-transport that process there inside there. Now there is a last part of this process of movement of food I want to talk about, and that is vomiting, because vomiting moves the food kind of unconventionally, and that is against this whole route. It's from stomach through the mouth. That's not a normal process of releasing the content. That's why it creates some issue in normal condition. Vomiting actually reflex, and that reflex is coordinated in the brain stem. It's completely coordinated in the brain stem over there. And this vomiting reflex can be stimulated by multiple factors that we know. It could be due to the excessive stretching of the stomach due to some reason, like the stretching of the stomach depends on physical activities as well as some emotional activities as well. Abnormal vestibular stimulation like seasickness. It could be urinogenital pain, it's kind of testicular pain or something like that. Any other kind of painful injuries can also allow this whole process of vomiting stimulate. It could be like increased intracranial pressure can also result in the vomiting tendencies, and tickling to the back of the throat, that most of us sometimes of our life actually get this idea. And noxious or any kind of toxic chemicals can also make it happen. Now vomiting is processed by the copious saliva production that start to build in our mouth. Once that copious amount of saliva start to produce and no much food to be present in our mouth, that is the condition that signals the nearby cells and the muscles to put all these things that are present in the stomach outside our body. And the sequential events of the vomiting follows reverse peristalsis movement from the small intestine into the stomach and through the stomach through the mouth. So it's kind of against all the odds, it's against all the whole process of digestion. And the stomach antrum contracts, forcing the stomach and intestinal contents through the lower esophageal sphincter. So even though the sphincter is present to block this backflow, but still at that condition the force is so much due to the contraction of the antrum of the stomach that will start allowing to throw all those components into the esophagus, and through the esophagus completely through the upper esophageal sphincter, and through that out of the mouth. Okay, but vomiting, though it seems bad, though it feels bad, as well as it's also like tiresome process after which we get very tiresome, but actually it's a kind of a protective function, protective process to remove harmful substances from our GI tract, from our body. That's why it's also very positive and important task to do when our body is compromised with some kind of bad substances in our GI tract. So that's all about the movement of food, both the way from mouth to the stomach and then stomach to the mouth as well. Welcome back, friends. Welcome to another part of the digestive system lecture. That's the third part of the digestive system, and here we are talking about the different types of digestive system secretions. There are different organs present in the digestive system. Those are made with different tissues and cells. They are destined to produce a specific type of chemical factors that helps in the process of digestion. Digestion deals with both secretions as well as absorptions. First, they need secretions to break down the food into smaller fragments and produce nutrients in the unit form. Then they will take those nutrients and absorb that. So digestion is destined and designed in such a way. Now let's look at the process of the secretion. And that digestive secretions involve the production and release of juices and hormones by the gastrointestinal tract and its accessory glands like liver, pancreas, gallbladder and stuff. The goal for our learning is to understand and list the secretion of the digestive tract, to describe their function, and to describe the control of the secretion with the help of both nervous system as well as hormone mediated system. The first slide that I want to talk about is more of a fact slide, where we want to say the typical daily consumption of food is approximately 800 gram for an adult, and fluid is approximately 2 liter. Now among that, about 1.5 liter of the saliva is secreted into the mouth per day. So just imagine those things, 1.5 liter of saliva, approximately 2 liter of gastric juices secreted every single day, and pancreas delivers more 1.5 liter of the pancreatic juice, while liver or gallbladder start producing the bile of 0.5 liter, that's a half liter, and small intestine produces approximately 1.5 liter of the fluid as well. So you think that this whole secretions, if we join them together, it approximately gives us around 9 liter. So it's a huge deal, right? So the gastrointestinal tract contains about 9 liter of the fluid every single day, and it's about only 0.15 liter is eliminated with the feces. So most of them are required for the conversion of the food that we take and then we ultimately absorb in our body, and which is very, very remarkable, and that help us to understand about how remarkable our digestive system really is. Let's move into the first kind of secretion, that is salivary gland secretions, sorry, should be gland or saliva secretion. The extrinsic salivary glands include three different types. One is a parotid gland, this is a pair of parotid gland actually, submandibular gland and sublingual gland. These are the three major types of glands that we have as a salivary gland. While parotid glands produce serous fluid, and that fluid contains enzymes, electrolytes and different kind of mucin, a little amount of mucin which helps in to produce mucus a little bit. That's why we have a kind of mucus around in our mouth. Submandibular gland and sublingual gland produces more of a viscous fluid than parotid gland. Okay, and all of them ultimately releases whatever things we call them saliva, that carries salivary amylase enzyme that has the capability to break down starch and polysaccharide. So that's why if you eat any carbohydrate food, there is a first kind of chemical reaction will take place in the mouth due to the release of all the saliva coming in from the salivary glands. So saliva provides plenty of functions. One of the major function is the protection of our mouth against different bacteria and other foreign pathogens, because they have an antibacterial molecule secreted known as lysozyme. It's present in our saliva. It also carries immunoglobulin A, that is an antibody targeted, and this immunoglobulin A is known as a secretory antibody because it's secreted with saliva, tears and stuff. So that helps in protecting our body. Saliva also helps in the process of taste because it will dissolve the food chemicals. Then it will also help in the lubrication, right, because it will also produce a mucus a little amount. And fourthly is the digestion that we talked about, where it can break down starch with the help of amylase. Now if you look at the regulation of the saliva secretion, it is regulated both with the help of mostly it's with the help of the nervous system. The whole process is regulated there. So there are two scenarios you can see here. One is the satiety and then the stress. So normally when there is a process of involving with eating a food, or you're seeing a lot of foods and stuff here around you and you have the access to all of them, so the parasympathetic nervous system of your body start producing acetylcholine. Acetylcholine helps in involved with some chemical signaling inside the cell that ultimately produces inositol triphosphate and diacylglycerol. Both of them are kind of, diacylglycerol is a part membrane bound structure that will ultimately allows the further downstream signaling, which helps in release of and fusion of the saliva containing contents and vesicles inside the cell, that allows the saliva to come out in your mouth. While if you are going through any kind of stress, then sympathetic nervous system will start acting and norepinephrine will be made, that will produce a lot of cyclic AMP, and once they produce cyclic AMP, that will prevent the release of saliva. So mostly the process of salivation is controlled by autonomic nervous system, both the parasympathetic and sympathetic, as we saw just now. And normally they involve with the nerve, facial nerve, that's nerve number 7, hope I am correct, and another one is the glossopharyngeal nerve, that is the nerve number 9 here, glossopharyngeal. Both of these nerves carry this whole process. This parasympathetic stimulation causes watery and enzyme rich secretion from the salivary glands. Normally the touch, sight and the smell of the food stimulates these salivary glands, and you can just take different experiments to prove that. We have plenty of experiments to prove that thing. Like if you are entering into a good restaurant with a very appetizing smell, you see the appetizing smell, that means that smell stimulates our sympathetic and parasympathetic, mostly parasympathetic nervous system influence to produce saliva. And acidic substances and pressures of chewing also cause the increase of the release of saliva, because if you put something in your mouth and start chewing it with your teeth, that will also allow to release more of a saliva from the salivary glands. On the other hand, if you have a stress, kind of fear or fatigue, or if you're sleeping in these conditions, that inhibit the process of salivation. Otherwise, just imagine the amount of saliva we produce while we are staying awake is generally not produced during sleep. But sometimes while you are looking through a dream or something like that, saliva sometimes come out during the sleep as well. And second thing, that is another very important kind of secretion that goes on mostly in esophagus, is known as secretion of mucus, and that's also we can call it as a esophageal secretions. And this is the only secretion that is made by the esophagus, okay, because this mucus is very important component, and they actually produce component known as mucin, which is converted into mucus. Mucus is a very viscous and mobile thing that will actually make this area of esophagus slippery, and it also coats the whole esophageal cells so that the food can easily pass through the esophagus. This is one thing. Second thing is that normally, esophagus is connected with the stomach, so if there is, stomach starts secreting acids in different contents, and there is a chance if stomach start to squeeze back and the lower esophageal sphincter is somehow releases, these acids can backflow into the esophagus. This phenomena is known as acid reflux, or it's also known as a disease called GERD, chronic esophageal reflux disease. That thing can cause the esophagus cells to kind of damaged by the activity of the acid, with the content of acidity is very high here in the stomach. So to prevent that, this esophagus produces those mucus that covers the whole esophageal wall and protect us against it. So this is how it look like. This is the intestinal fluid, these are the cells, and this will start producing bicarbonate to fight against that acid as well, because bicarbonate as well as they start to neutralize the environment of pH, because stomach lumen have a pH around 2, which is very acidic, like the hydrochloric acid and sulfuric acid at that range. Normally they produce hydrochloric acid, but that's very acidic, this pH 2. So once they produce, this mucosal cells arranged to have a based on pH 7, and they start secreting those things that to protect us against it. Now if you go to the third kind of secretion, it is a gastric secretion. This is one of the major secretions of our digestive system. Gastric secretion is mostly it's called a gastric mucosa. Those are the cells that are present there. They start producing different chemicals with the help of exocrine and endocrine as well as paracrine secretions. The exocrine secretions collectively called the formation of gastric juice. Approximately 2 liters of this gastric juice is produced every single day, and it includes mucus. So generally gastric juice has several constituents. I say it has mucus, it has HCl, it has pepsinogen. Pepsinogen is a type of enzyme that can produce pepsin. Actually pepsinogen is a precursor of the enzyme pepsin, which is a protease enzyme, and they start to produce several type of intrinsic factors which has functions like mucus throughout the stomach is produced, and it's protecting the lining of the stomach cells. Pepsinogen is also secreted throughout the stomach. HCl is produced by fundus and body. These are the two parts that produces HCl through this place. Okay, and intrinsic factors are also produced by fundus and body. Okay, but rest of the parts produces rest of the body place. And this is signaling process known as enteroendocrine signaling, because if you look at the cells now, if you zoom into the structure, you'll see these are the zoom in structure. This is another same kind of structures we are observing here. Here you see different cells like G cells are present. These are the G cells. There are also chief cells, there are parietal cells, there are mucus cells. Different cells are present, and some of those cells known as enteroendocrine cells. Those enteroendocrine cells that are mainly present in the pylorus region, these are where the enteroendocrine cells are present, they start releasing gastrin. Gastrin is a hormone, and that's why we call these cells enteroendocrine hormones, enteroendocrine cells, because those are endocrinal secretions and secreted by the cells that are present in the enteric body. And here you see that all of them, right, chief cells, mucous cells, they start releasing components and factors inside by that fashion. The specialized cell produce each of these gastric secretions separately. For example, the stomach mucosa is invaginated, if you look, because you know this whole stomach body, this whole cells are invaginated and they form deep wells they are called as a gastric pit, and they are filled with gastric glands, as you see here. If you look at the fundus, this area, you'll see they start forming these invaginations, and one of such invagination is this one forming called gastric pit and gastric glands. Gastric glands produces mucus, because if you zoom into one of the gastric glands, it has mucose cells, they produce mucus. Gastric glands have like in the body as well as in fundus, they also produce HCl, and there are two types of mucus that are produced in the stomach. One is the thick alkaline mucus, another one is thin watery mucus. The parietal cells, if you look at here, these are the parietal cells, the specific design cells in this picture. These parietal cells, they start producing HCl and intrinsic factors, while mucous cells producing mucus. And there are other cells known as chief cells. These pink colored cells are the chief cells. These chief cells, they are also known as zymogenic cells, produce zymogen, which is an inactive proteolytic enzymes, okay, and that will help to produce pepsinogen. And some cells in the gastric glands of the pylorus, if you look, they have a G cell. G cells mainly present in the pylorus, they are enteroendocrinal cells, they produce gastrin. Okay. Although all of this, most of the secretions are made from the stomach cells and the gastric pit and gland that is formed inside the stomach, but we still keep another slide for explaining the stomach secretions and some examples of that as well. Normally, you know, we know that stomach produces mucus, it produces HCl, intrinsic factors and stuff. The mucus that stomach produces, that protects the stomach from the self-digestion, that's also known as auto-digestion by all those neutralizing stomach acids that's present there. So here this mucus actually neutralizes those stomach acids, and also it inhibits the activity of the protease known as pepsin. While we have mucus and the tight junctions between the cells in the gastric mucosa, they are collectively known as the gastric mucosal barriers, because if you look at the cells that are present and making the stomach, it is not loosely placed like that. We have a tight junction formed in between the cells so that all those acid contents and all those components cannot take entry in between the intercellular junctions there. Okay, only aspirin and alcohol are absorbed across the stomach's mucosal epithelium. Otherwise, rest of the things don't have any entry point there. When aspirin or alcohol passes through the stomach's mucosal barrier, they can destroy those cells. That's why ingesting a lot of alcohol or aspirin day by day can harm your stomach, in turn harm your digestive system. And the HCl that is secreted by the stomach, especially the parietal cells, that lowers the luminal pH between approximately 1.5 to 2, this pH contents that is produced. Mostly it's produced by fundus and the body. You see the proton content is listed here. And in this case, this change in pH, like on the scale that represents 10-fold change in pH in the stomach, for the example of 2 pH, that is compared to the mouth, which is 7. Sorry, it's 2 pH compared to the mouth, that is 7. So imagine how much acidic the content will be here. And actually the release of HCl is not to digest a food, it's actually to make other enzymes activated and that high acidic content. For making all those proteolytic enzymes active, they need to release this acid. The highly acidic gastric environment is also very lethal for most of the bacteria and other microorganisms. So we won't find generally bacteria there in the stomach until and unless there is any compromisation in the lining production in the stomach. And if this compromisation is done due to the taking of aspirin or taking of a lot of alcohol day by day, or due to any kind of ulcer formed there, then bacteria can take place or take entry through that place. Or there are few bacteria like Campylobacter jejuni, they can actually present there in the stomach and they can actually be there beating this acidic content over there. So that's one kind of bacteria that can colonize in stomach even though there is a high acid content, but most of the bacteria cannot. The HCl functions here to break down the plant cell walls, most of the connective tissues if we eat the plant product, because we used to eat a lot of plant products there. It denatures the protein a little bit, but mostly it converts that pepsinogen, which is the precursor molecule of pepsin, into the active pepsin. Once it produces active pepsin, that is the active enzymes to break down protein components, right? Now the parietal cells start secreting another factor known as IF, or intrinsic factor, which is also required by the intestinal cells to absorb vitamin B12. If we don't have intrinsic factor released properly in our stomach, vitamin B12 absorption will be halted. Because if the vitamin B12 content is halted, if we can't absorb vitamin B12 in our body through stomach, that means our RBC production will be compromised there, because production and maturation of RBC required a lot of vitamin B12, because it's a cofactor. This acts as a cofactor in the process of the RBC maturation, and that can cause anemia known as pernicious anemia. The peptides in the stomach triggers the release of another hormone called gastrin, that is from this pyloric region, and these are the cells present here known as the G cells, through where all these molecules, all these hormones start to release. And they also, this gastrin stimulates the HCl secretion by the parietal cells, and also the histamine secretion by the paracrine cells that is present here. So paracrine cells also produce these histamines, and histamine acts as a kind of it synergistically act with gastrin to stimulate the release of HCl. So it's kind of a positive feedback loop going on there. Once it produces this gastrin from the G cell, it start producing more histamines from as well. So histamine and gastrin together help producing more and more HCl in the stomach to make the content more acidic and keep the content acidic over the time. That's the idea. Now if you go to the other type of secretion made by pancreas, it's known as the pancreatic secretions. Pancreatic secretions mean they produce a lot of juices and chemical factors together, known as pancreatic juice. Now most chemical that produced by the pancreatic secretions, they are required in the digestion process, and mostly they are proteolytic, and also a few parts they also produce lipolytic enzymes as well, but very less, mostly proteolytic. The secretions that initiate the process of chemical digestion in the intestine actually comes from the pancreatic juice, okay? And in this case they produce some important facts. One of such important fact is bile, that produced by the liver mostly, but it is stored and released from the gallbladder, that emulsifies fat to increase their surface area, and subsequent chemical digestion will take place. While the exocrine pancreas produces two types of pancreatic juice: enzyme rich pancreatic juice, that is one thing, enzyme rich, and second one is bicarbonate rich. So one of such juice contains a lot of enzyme factors, one of such can contains a lot of bicarbonate factors. Both of them are required in different purpose. The enzyme rich pancreatic juice is stimulated by the hormone cholecystokinin or CCK, while the bicarbonated rich pancreatic juice is stimulated by the secretion of secretin. Okay. Now the exocrine pancreas secretions are often delivered through the different sort of like hepatopancreatic sphincter that is present there between the pancreas and the junction between the duodenum. So that sphincter regulates the release of those exocrine materials, and they follow with some of the enzymes. If you look at the enzyme rich secretion, they contain some of the enzymes. One of such is like mostly proteases. Example of such protease is chymotrypsin, produced by the pancreas. Second type is amylase, that's known as a pancreatic amylase because it's secreted by pancreas. The first sort of amylase released by our salivary gland is known as salivary amylase, and it also produces lipase or lipase, whatever you say, the enzyme to break down lipid. Now the action of lipase will only be strong when there is a release of bile from the liver, otherwise lipase cannot act on large fragments of fat. Now pancreatic proteases includes chymotrypsin, trypsinogen and procarboxypeptidase. All these things, chymotrypsinogen, trypsinogen, whenever we have a gen attached as a suffix there, that means that chemical or that enzyme is not properly active. These are the pre-molecules. We need to cut, and like some sort of modifications are required to make them active. So let's say trypsinogen is converted into trypsin, chymotrypsinogen is converted into chymotrypsin, and procarboxypeptidase is converted into carboxypeptidase. And actually enterokinase is the enzyme that helps in the conversions of all those trypsinogen into trypsin, chymotrypsinogen into chymotrypsin and stuff. Once they produced, mostly like say the enterokinase enzyme that converts the trypsinogen into trypsin, enterokinase, that's the enzyme that converts chymotrypsinogen into trypsin. This trypsin activates chymotrypsinogen into chymotrypsin, and then trypsin activates procarboxypeptidase to produce carboxypeptidase. So these are chain events, but first started with enterokinase enzyme. Okay, and there are also a different set of cells known as duct cells that are also present inside this pancreas. Those duct cells start secreting bicarbonate. That's the second part of the secretion, bicarbonate rich secretion. This bicarbonate rich secretion from the duct cells to neutralize the acid from the stomach and to produce an optimal pH in the duodenum so that the rest of the process can work finely. Because stomach is designed in a way to support and live in the acidic environment, while the duodenum cannot receive that much of acid and live healthy. So duodenum needs to neutralize all those components, and that is done with the help of the secretion of bicarbonate there. Okay, and the endocrine pancreases, that's the process of endocrinal signaling. It also secretes some hormones because that's also famous for secreting hormones. Two such hormones secreted by pancreas, one is insulin, another one is glucagon, which are required for the metabolic pathways of after the digestion event, but not exactly in the digestion event. While insulin regulates the absorptive state and glucagon regulates the post-absorptive state of the process of the digestion, but mostly controlling the glucose concentration in the bloodstream, and that's an important role to play. The regulation of pancreatic secretions are made up with different hormones like secretin, GIP and CCK. Secretin says like acidity in the intestine causes increased sodium bicarbonate release to neutralize the environment. Fatty acids and sugar causes the increased insulin release, and CCK, fats and proteins, cause the release of this CCK. So all these things, depending upon what kind of content that we have and that we are digesting, stimulates the release of all those different hormones. And here is a feedback loops presented, a positive loop and a negative loop, where it's controlled by hormones as well as parasympathetic nervous system. Now it's time to look for the secretion comes from liver. And when you talk about liver, I always get fascinated about the function of liver, because this is one of the most important organ of our body, because it carries a lot of functions for metabolism as well as in digestion in so many different directions of our body, to release all the toxic materials from our body and everything. So this is kind of a hero organ of our body. The main digestive function the liver plays is to produce what is called as bile, and bile is associated in the digestion of fat. If they don't secrete bile, the fat digestion will be kind of impossible. The bile backs up into the gallbladder, so it's secreted by liver, and through this duct it will go to the gallbladder through this systolic duct, and finally those all ducts, common hepatic duct and systolic duct, they all tie together to finally form what's called a common bile duct that will ultimately release all those components into the duodenum. And they have here what is called hepatopancreatic sphincter just between this duct end, where we have the connection between the bile storage site, that is gallbladder, pancreas and the liver. So three different ways connected to one single common duct. There is a sphincter to regulate this whole process, that is hepatopancreatic sphincter. Now the two major components of bile is organic compounds and bicarbonate solutions. The organic compounds that we have mostly are bile salts, that helps to emulsify the fat. So if you have a large fat component, it will break those components down into smaller chunks and fragments, so that all those lipolytic enzymes have much more surface area to work and break down those fat components into further smaller fragments, right? And bile work associated with the pancreatic lipase, which comes through the pancreas. So once bile fragmentize and emulsifies those fat, then lipase will act and break the fat down into fatty acids and glycerol. That's how the digestion takes place there. Okay. And it also contains lecithin, it contains cholesterol, and it contains bilirubin. So bilirubin, lecithin, cholesterol and all the rest of bile salts are secreted there. While cholesterol and bilirubin are eliminated through the feces, but bile salts are recycled throughout the time, because they are required all the other times in different process. They are recyclable, and they stimulate the secretion of bile from the liver, and then those bile can store into the gallbladder. So that's why we have a storage site, because liver is the busy organ, it cannot produce those bile wherever it's required in their process. So whenever you digest food, some part of the bile which is stored in the gallbladder, that will come to help in the process of digestion. So when we stimulate this whole process, like when you stimulate the release of these chemicals by the enzyme, by the hormone actually secretin, that's called as nature's antacid. Secretin is secreted. Bile duct cells secrete bicarbonate solution that is identical to the pancreatic bicarbonate solution as well, and that function as a liver neutralizing agents. So here liver is not only allowing the fat digestion, but also it starts secreting all those enzymes. It starts release that secretin, and secretin, remember this hormone known as a nature's antacid, because it's naturally protecting us against acidity, because it's a bicarbonate solution that's produced. Now it's time to look at the secretions made in the small intestine. Small intestine have the submucosal glands, it has the Paneth cells, it has the goblet cells. All those different types of cells and tissue have different secretions. While the goblet cells release mucus, Paneth cells produce enterokinase, which is an enteric kinase enzyme that activates all those pancreatic zymogens, and submucosal glands produce viscous mucus to prevent the damage of the mucosa. Okay. If we look into little deep, we have those intestinal glands, or also known as crypts, intestinal crypts. Here you can see the detail of those such of a intestinal crypt, where you have a brush border actually made up with the villi kind of cytosolic extensions of those cells in the intestinal epithelia. It's like a brush border because those are the microvilli of the cells that we can see. And the concept of villi and microvilli is that normally the cell lining should be like this. Once they form villi, it start to form like this. Once it form microvilli, it actually start to form things like this. So start to think of why we need to do these things. This is without any villi, this is with villi, without microvilli, this is with microvilli and villi. So the more we are dividing, that means the more surface area is increasing, and the job of the small intestine is the digestion as well as absorption of the food. That should be placed properly here. So that's why we have a lot of microvilli contents produced in there. Okay, and those cells, those epithelial cells, they produce certain enzymes. Obviously that's one part of the job, but they have another job of absorption. So that's why they need to keep in mind to modify all their components so that they can interact and involve with this whole process. Lastly, we have a large intestinal secretions. Large intestinal secretion, they have alkaline mucus in the large intestine containing bicarbonate. The mucus that we have in the large intestine, we have a bicarbonate, we have potassium ion that's secreted by the intestinal cells, because both these potassium ions that is secreted as well as this bicarbonate helps in the neutralization from the stomach acids and chyme. Potassium also helps in the process of preventing any kind of bacterial acids and damage. The role of mucus in the large intestine is much bigger compared to the any part that mucus is present, because the mucus in the large intestine coats all the mucosal cells, all the epithelial cells, that the first hand in the first order is coating all the cells, so it will prevent any sort of acidic damage. It will also prevent any sort of pathogen to enter, because this is large intestine and this is filled with E. coli. It's a healthy and friendly bacteria of human body that resides there. Not only E. coli but multiple bacteria resides there in the intestine, and the role of them is to produce vitamin K, and they also fight against some other invaders or foreign pathogens, right? That's why we need to take a like close look of gaining a good healthy gut microflora is a better way of living healthy life. That's why we involved with the idea of probiotics, where we take all those healthy bacteria from outside with the help of like yogurt and other stuff, so that it can keep us healthy. Now the mucus that we see here, it lubricates the feces and protects the large intestinal wall from any sort of mechanical damage and friction that is generated during the release of the feces. Let's say a person is not drinking adequate water. In that case, the feces becomes hard and very, very hard and strong. That can lead to the damage in the intestinal wall, that can ultimately lead to the release of blood and stuff thereover. That can be prevented. And both the reflex that is required here, long reflex as well as short reflexes that are triggered by the mechanical stimulation of the acid and the increased secretion of alkaline mucus from the large intestine start to release. So the more friction it will be generated, the more feces are going to involve with the interaction with the wall, the more they want to create mucus, so that those feces will remain soft and it can release without harming any of the cells and intestinal lining of our body. That in a sense are different sort of secretions that are required in the process of the digestion. We've been talking about digestive system and the different parts of digestion in human body. In this video lecture, we'll be talking about the digestion and absorption that is going on in our body. We've been looking at like different anatomy of our digestive system. We saw the different types of control and regulation of the digestive system. Now this is the time to actually understand how the digestion and absorption take place and what are the different regions for this process. Now chemical digestion involves breaking down of the food with the help of enzymes, and those enzymes will break down macromolecules into small fragments and nutrients. Once those small fragments are generated, then they are absorbed, okay, in the GI epithelium cells. In this lecture, we will be discussing the action of digestive enzymes involving amylase, proteases and lipase, along with the function of bile salts. We will also describe the transepithelial transport of simple sugars and amino acids. And thirdly, we will be discussing the emulsification and chemical digestion and absorption of lipids, which is a slightly unique compared to the carbohydrate and protein digestion and absorption. Let's talk about the chemical digestion process that actually take place in mouth from the beginning till the end in the intestine. The ingested food first is broken down mechanically in the mouth. That's known as the mechanical digestion. Once they are broken down into fragments, into smaller pieces enough to be swallowed, then they will be taken inside the esophagus. Through the esophagus, they ultimately take entry into the stomach. Now inside the stomach, there are two types of digestion take place. One is a little bit of mechanical digestion and a little bit of chemical digestion as well. The small particles are then mixed with acids, which is hydrochloric acid secreted by the stomach cells. And then this acid will help and allow to break down some protein fragments inside. So this is a kind of protein digestion begin in the stomach. Now the major nutrients must be broken down enzymatically in sequential stages in the small intestine to finally produce the fragment portion of all carbohydrates, proteins and lipids. Okay. Now if you look at here, there are chain of all these things, polymers of carbohydrates, lipids and proteins. The polymer of carbohydrate example is starch. When broken down, it produces unit sugar, that is glucose. Similarly, we have protein molecules. When you break them down, it produces amino acid. And we have fats. When you break them down, we get fatty acid and triacylglycerol as a unit component. Here you see the different list of enzymes that are involved with the substrate breakdown into small fragments. For example, breakdown of starch is mediated by amylase, and it's secreted both one in mouth and another from the pancreas, and in both of them break them down and form maltose and glucose. Proteases act on stomach and small intestine, break down protein into amino acid. Lipase acts in the small intestine, in the duodenum, break down fat into fatty acids and glycerol. Now all carbohydrates must be digested enzymatically into the monosaccharide components. That's the first and foremost thing. Whatever carbohydrate we are dealing with, it must be broken down into the monomers. Now starch is digested into the monomer glucose, while sucrose is digested into the monomer glucose and fructose, and lactose is digested into glucose and galactose, while maltose is digested into 2-glucose. Okay, this should be produced. Proteins all the time should be converted into amino acids. Now the amino acids that they can produce by breaking down protein could be like a dipeptide, breakdown of this protein into 2 peptides or 2 amino acids together, or they can form tripeptides, so 3 of the amino acid can be linked. And most of this fat is broken down into fatty acids and triacylglycerol that we talked earlier. Now there are some also non-polar products in the digestion, and that can be absorbed by simple diffusion process, which is a passive diffusion through the phospholipid bilayer of the intestinal epithelial cell. Okay, and most of the polar substances are absorbed using active transport, or in some cases passive carrier mediated transport through the cell membrane. So we have the channel proteins through which it can take entry. Fats or non-polars can easily take entry through the membrane because that is mostly hydrophobic. Let's first talk about the carbohydrate digestion in the mouth. It's the first start point, and the carbohydrate digestion begins in the mouth. This is the type of chemical digestion that we're talking about. Normally, mouth is involved with two types of digestion. One is mechanical, second is chemical. In the mechanical digestion, teeth is involved to break and grind all those food into smaller particles. And then they will start mixing these food particles with saliva. Saliva contains amylase, and the amylase can break down starch into fragments. Amylase break down starch into following components. It can break the starch into maltose and maltotriose and some amount of dextrin. So maltose is normally a disaccharide, maltotriose is a trisaccharide, and dextrin is a kind of branched fragment that is generated. As you see, the starch is a hugely branched molecule. It's been broken into all these smaller fragments there. Okay, but when all this mixed amylase and all the chemical agents are transferred from mouth through the esophagus into the stomach, inside the stomach acid deactivates this salivary amylase. So the function of salivary amylase is only active in the mouth and when the food is moving through the esophagus. That's the only part salivary amylase is active. But once it's mixed, it is enough to break carbohydrate down because it takes less effort to break and digest carbohydrate. The second thing is about the protein digestion, and the protein digestion begins in the stomach. So most chemical process, like normally the process of protein digestion occurs in stomach because stomach releases acid and another enzyme known as pepsin. Actually, pepsin require acidic environment to act and break down protein components. That's why they need to release acid there, and they release hydrochloric acid in the stomach. While the pepsin will start activating, it breaks the peptide bond between the tyrosine and phenylalanine. Remember, it breaks the bond of any amino acid sequence or repeated peptide between tyrosine and phenylalanine into smaller amino acids, and they produce peptide fragments, and that contains few amount of an amino acids. So this is a larger protein broken down by pepsin into smaller fragments of amino acid chain. Stomach also responsible for a little bit of mechanical digestion, where the stomach has its own peristaltic waves generated that can break and grind foods a little bit inside the stomach. Now if we talk about the process of digestion in the small intestine. Small intestine is the region where most of the chemical digestion take place, and it is also involved with the absorption of all those nutrients inside. Pancreatic digestive enzymes alone are much sufficient to digest carbohydrates and proteins that are present here in the small intestine, because pancreas secretes all those proteolytic enzymes. Example, trypsin, chymotrypsin. These are the examples. It produces pancreatic lipase. They also provides pancreatic amylase. Pancreatic lipase is essential for the bulk of fat digestion, and for example, cellulose and other type of plant complex carbohydrate cannot be digested by the humans because we don't have any of the enzyme to break them. That's the reason we cannot eat grass, because the most of part of the grass is made with cellulose, which is the plant cell wall component. So indigestible plant polysaccharides serve as an important source of dietary fiber, which is also required for a proper movement of the bowel through the large intestine. And certain fiber type help the movement of the stool through the colon. Now if you look at here, the vegetables and the fruits contain this amount of dietary fiber, but they mostly contain valuable vitamins and minerals as well. So all those vitamins and minerals are absorbed in the small intestine. And most of the dietary salt that we take, most of the dietary salt that we take, let's assume these are all dietary salts, those things are absorbed by the small intestine by either the transcellular or paracellular transport mechanism in the small intestine. So what are those type of scenario? In the transcellular transport of water, water is linked to active transport of sodium via sodium potassium pump. So water is taken and dragged inside with the help of that sodium potassium pump. Sodium is actively pumped out through that process. So let's say this is inside, this is outside. The sodium is dragged out, potassium is dragged in. Once the sodium is actively pumped out of the digestive tract at the basolateral membrane of the epithelial cell, the sodium then moves passively from the GI lumen into the epithelial cell through the ion channels, which is a sodium glucose co-transporter channel. So once sodium is moving inside the cell, glucose is also moving inside the cell, and then chloride follows the sodium. It's through the electrochemical attraction process, chloride also start to flow inside, and then as a lot of ions are moving inside the cell, the concentration of ions are increasing inside the cell. And to keep this concentration of the ion concentration inside and outside constant in an equilibrium, water start to passively inside those cells via osmosis. That's the overall idea of absorption that is taking place. As you see here, we will be looking at this process, that this is the first part of the digestion in small intestine, mediated for the carbohydrate digestion and absorption. So here come the pancreatic amylase that continues to digest all those large fragment of carbohydrates into smaller fragments, and actually these enzymes work in the duodenum, which is the start point of the small intestine. And there are intestinal cells known as the brush cells. So this is known as the brush border of the cells. Okay. For the digestion, these brush border enzymes complete the process of carbohydrate breakdown into monosaccharides. And in this case, normally the glucoamylase break down the maltose. So there are different enzymes. Glucoamylase break down maltose and maltotriose. So those things are broken down by glucoamylase. Dextrinase breaks down dextrine. Okay, I'm not writing it here. So dextrine is broken down by dextrinase. Okay, any enzyme you'll see ase will be the enzyme to break it down. For example, to break down sucrose, we have sucrase secreted there. For breaking down lactose, we have lactase. So these are all carbohydrate breakdown enzymes that are secreted there and work there. Now most nutrients that are absorbed mostly by the transepithelial transport. So what is that process? You know, there are different transporters that are present in the epithelial cell membrane. Normally it could be primary or sometimes secondary active transport. In the secondary active transport is which the glucose and galactose are co-transported. Glucose and galactose are co-transported inside the cell together. Fructose enters the epithelial cell normally on its own via the own transporter that we see. It's a fructose specific transporter, but glucose and galactose, they transfer the same time at a specific region. And we also have glucose sodium co-transporter, which also helps to move glucose using the sodium ion concentration gradient there. And once inside the epithelial cell, all the monosaccharides that take entry, monosaccharides exits the basolateral side of the cells in the same type of transport molecule via the facilitated diffusion, because that will be possible to move from there into the capillaries and spread through the body and rest of the area of the body cells. Here you see the process that once this glucose and sucrose and fructose, all these things are entered inside the cell, they will be transported out into the capillary by the process of facilitated diffusion, where there is a carrier protein required to transfer those monosaccharides into the specific place of the capillary. Now the second part of the digestion in the small intestine is the digestion of proteins. And if you look at the digestion of proteins here, if you see, there are small intestine, because in the smaller intestine we have the protease enzymes released from pancreas, a variety of proteins and protein breakdown products that are formed. For example, trypsin. Trypsin is an activated form of trypsinogen, while chymotrypsin is an activated form of chymotrypsinogen. Carboxypeptidase is an activated form of procarboxypeptidase. They are all the major pancreatic proteases that approximately work at pH 7 for the process of protein digestion. Trypsin and chymotrypsin, these both two, trypsin and chymotrypsin, split the protein into smaller peptides and some of them into single amino acids even. So only single amino acids can be generated. While carboxypeptidase splits single amino acids into carboxyl end of the protein and start breaking down amino acids from the terminal. So carboxypeptidase work after the activity of trypsin and chymotrypsin. Okay, and here you see there are also brush border cells that are present in the epithelium, right? So these are the brush border enzymes known as aminopeptidase and dipeptidase continues the process of digesting those fragmentized amino acids so that we can absorb that. So once they produce either they can produce single amino acid or produce di amino acids, or they can produce tri amino acids or tripeptides that can be absorbed through the small intestinal cells. A number of different amino acids are co-transported into the luminal side of the epithelial with the help of sodium and via secondary active transport. So again, sodium can help, the sodium gradient across the cell, let's say a lot of sodium gradient is generated here. Sodium gradient across the cell can help to bring some of this peptides as well, dipeptides and tripeptides as well, to bring inside the cell as a co-transporter mechanism. Now most of the proteins absorbed as dipeptides and tripeptides, as we talked earlier, and which are further digested into single amino acids inside the GI epithelial cells. So once you get a dipeptide or let's say tripeptide inside this epithelial cell has a lysosome that is filled with proteolytic enzymes. Once they release that, it can break those tripeptides and dipeptides down into smaller single monopeptides. Now if we have more hydrophobic amino acids, and those amino acids tend to leave the basolateral side of the epithelial by normally simple diffusion mechanism into the capillary there. Now if you have more hydrophilic amino acids, they want to exceed the GI epithelial by the process of facilitated diffusion. So in this picture it's shown that mostly these amino acids are hydrophobic, so they can easily transfer themselves through the membrane into the blood vessel there. But if it carries more of a hydrophilic one, it should have a carrier protein embedded into the cell membrane that will help to move the amino acids into the place. Now it's time to talk about the digestion of fat in the small intestine, and fat digestion is kind of unique. It's slightly different compared to the digestion of digestion of carbohydrate and protein, because fat digestion takes a lot more time. Most of the dietary fats are triglycerides, which are made up of 3 fatty acids. It could be like short, medium and long hydrocarbons, and each of them are bonded with glycerol, okay, with the help of ester linkage, those fatty acids. So there is a glycerol and hydrocarbon tails are attached to it with the help of ester linkage in the middle. Now all of this actually most of this digestion occurs in the duodenum of the small intestine. Mostly the process of chemical digestion that conveys in the small intestine occurs in the duodenum, and the absorption takes place in the further downstream of the small intestine. Okay, now there are 2 mechanisms act to increase the surface area of the fats and subsequent chemical digestion. One event is the release of bile, and second event is the segmentation, which is a physical event as well, segmentation. segmentation in the small intestinal wall. Segmentation will help the fat to disperse into the aqueous layer, like the oil and the vinegar. If you see here, this is the oil and vinegar and how it's dispersed. If you have a segmentation, like as you see, there is a segmentation here in this specific bottle. Due to this kind of segmentation, we have a different way of separating the oil from the rest of the part. So most of the molecules that are present on top are the oil, are the fat, and most of the aqueous solution is residing in the bottom. And if you do the same thing with other kind of structures, it will be kind of hard to achieve, but if you have segmentation, it helps. And the second thing, bile. Bile salts attach to the smaller fat droplets and prevent them for attaching with themselves and form a large fragment. So it will keep this smaller fragment of fat separated, and we call it as an emulsification. Now, as the bile salts are amphipathic in nature, that means they have a hydrophilic as well as a hydrophobic side, they can surround the small lipid droplets. So let's say this is a small lipid droplet, and the bile salts are surrounding that lipid droplets and separating it to the rest of the other lipid molecules, okay, and prevent them from coming together. So in this case, they increase the total surface area of each of the lipid fragments by separating them into smaller fragments. So increasing in the surface area of the lipid helps for more efficient hydrolysis of these lipid molecules by the pancreatic lipase enzyme. So pancreatic lipase will now act, right? So lipase digests triglycerides into monoglycerides and free fatty acids. So what it will produce? It will produce the glycerol. That backbone will be there, the glycerol backbone, and we have all those hydrocarbons released. Now that backbone can also be broken into smaller fragments. So it's a triglyceride, it can break them down as well. Now bile salts also surround these monoglycerides and free fatty acids as well to form what tiny droplets known as micelle droplets. It's kind of like 1 million times smaller than the emulsified fat droplet, very, very small. So even after the digestion is done, still the bile salts keeps like circulating, keep covering those fragmentized portions, okay. And why it is required? Let's say here this is the large lipid droplet, mixing of bile salts, breaking them down into smaller fragments known as micelle, and micelle is formed even after the breaking of the fragments. We have those bile salts to cover them up. That's what we call micelle here. Now once we form the micelle, it comes close to the luminal membrane. This is the luminal membrane that we can see, close to the luminal membrane of the epithelial cells here, and they release their monoglycerides and free fatty acids inside the cell via simple diffusion through the lipid bilayer. That's what we can see here. So here we have the smaller fragments of fatty acids and monoglycerides that can be easily diffused through the core area of the cell membrane, because most of the part of the membrane is actually made with hydrophobic region. So now this is one of the way of delivery. Or what happens, some of this micelle can directly interact with the cell membrane that ultimately release triglycerides and fatty acids inside the cell. Once we need to transfer that triglyceride to some distant area of the cell for the process of metabolism, that triglyceride and those fatty acids are further coated with proteins, and then we bring them to the capillary or any lymphatic system if we require. And those fragments which are coated right after the process to transport are known as chylomicron. Chylomicrons are the dressed up fatty acids and glycerols that is transport ready, okay. And this chylomicron exit the basolateral surface of the epithelial via exocytosis process, and then they will enter the lacteal lymphatic system here. So this is the lacteal lymphatics. Through this lacteal lymphatic system they will be migrated. They don't actually enter into the capillary, they go to the lacteal lymphatic system. So that's how the release of fatty acids are made in the digestion in the small intestine. Now the last thing that I want to talk about is the absorption process that takes place in the large intestine. because large intestine is not responsible for the chemical digestion, neither it's a function of secretion. So the only function it plays is the absorption of the nutrients and absorption of water and absorption of some amount of ions and vitamin K. So there's a kind of symbiotic relationship between some bacteria that resides in our large intestine. Some of the example is E. coli, that's a friendly bacteria that is present in our large intestine that helps to generate vitamin K that is absorbed by these large intestinal cells, as well as large intestinal cells absorb a lot of water to make the feces much more concentrated to be released outside the body. So here the byproducts of those colic bacteria metabolism include small amount of vitamin B complex and a huge amount of vitamin K. Both are absorbed in the intestine. And here vitamin K is very essential for producing blood clotting proteins. So without vitamin K, the blood clotting pathway can be compromised in our body. Now the colon absorbs the small quantities of salt as well, as you can see here. This is lumen of colonic crypt and this is the interstitial fluid. So what we are going to do, it can take some amount of sodium and chloride ion inside. Chloride ion enters into the lumen with the help of CFTR channel proteins present. Sodium is reabsorbed, and it also, as they start to build up a lot of sodium and potassium inside the cell, it will automatically allow the water to backflow to balance the osmotic pressure. That's the whole idea of the absorption there. It's not a primary content of absorbing water, but as a lot of sodium and chloride is being absorbed to balance the equilibrium of the concentration of the ions, water flow in the osmosis pathway. That in a sense is the absorption process that takes place in the large intestine. I hope this video helped you to understand about the digestion and absorption that is going on in our body. So if you like this whole series of video about the digestion, please hit the like button, subscribe to my channel to get more and more anatomy and physiology videos like that. Thank you.